All right, last lecture for the class. This is chapter 26, part 2, where we're going to talk about environmental influences of gene expression. So there are internal and external environmental factors, variable expressivity is variations in degree to which an allele is expressed in an individual, meaning just because you have a trait does not mean it's going to be strongly expressed. There is a process in the DNA where they're in the DNA, in the passing on of the DNA from the parental alleles, where you have some kind of random variations which can occur, which make you unique and not carbon copies of your parents. Lack of penetrance is where the allele is not expressed in the population. And so you may have a suppression due to the fact that it's not going to show up in the population, even though it is there in the allele. So, for example, calico cats are female. You do not see a calico cat that is a male, so it's not expressed in that particular population. Okay, so the central dogma is kind of what all of biology is based on, and it includes genetics, and so that's why we're talking about it right now. So DNA is able to do the following: make copies of itself. When we make copies of it, it's like when I make your guys' test, I print one off my computer and then I go and make copies, and each and every copy of the exam should be exactly the same as the original. So this is in preparation for mitosis and meiosis. So when cells divide, the DNA has to also be replicated. It doesn't get divided into smaller pieces. It gets replicated and then the copies get shared on. DNA can also mutate. This is chemically changed, and then those changes are passed on. So mutations can be passed on. Mutations can be acquired or inherited. So mutations as far as like if you're exposed to an environmental factor like radioactivity, toxins, poisons, things like that can be passed on after the DNA is mutated. DNA is also able to store information determining characteristics of all cells and organisms. So this is where our genotype is stored. So if we look down at this bottom image, DNA is in the box. DNA can go to the left and be replicated to make other DNA. But DNA can also undergo a process called transcription into RNA, which then undergoes translations into proteins. And the proteins can be structural, meaning you think back to our cells where we talked about the tubulars, the things that act like rebarb or steel to help support the cells and the walls. They can be carriers, so they can help in active transport processes, or they can be enzymatic or hormonal, so they play a role in our biological processes. So the nucleotide sequence is the gene. It's the blueprint for the synthesis of structural and regulatory proteins, so that's that very last group right there. And so what we didn't complete on the last cycle was those proteins then are the enzymes that are involved in replication, transcription, translation. So without the proteins, your body would not be able to replicate it. Your body would not be able to replicate its DNA. It would not be able to replenish the protein supply. And so DNA is going to supervise manufacture of RNA through the process of transcription. And when we have an RNA strand, it is a complementary copy of DNA. It's not an exact copy, like making a copy off of a printer. It's kind of like a translation. So going from one language to another, there are going to be some variations, but in general, the principal idea is still there. Then the RNA is going to undergo a process called translation to produce proteins, which are used for enzymes. So all of your enzymatic processes, if you're lactose intolerant, you lack an enzyme called lactase, and that is a function of not having the protein synthesized to digest the lactose in your body. Some enzymes are required for transcription and translation to take place. So like we said, those proteins that are synthesized from translation then feed back into the cycle to replicate, transcribe, and translate. So it's one big complete cycle. So a nucleic acid is the large polymer made up of individual monomers. So these are like Legos. If you've ever played with Legos, you know you have teeny tiny Legos. You can stack them all together and you can make a really big something like a big castle. So monomers are the individual Legos, and the castle is what we call a polymer, Polymer. And the individual Lego in DNA is called a nucleotide. The overall DNA or RNA structure is called a nucleic acid. So the nucleotide is going to be a 5-carbon sugar molecule, a phosphate group, and a nitrogenous base. Now the nitrogenous bases are important because of the pairing, so you do need to know the pairing, okay? So in DNA, make sure you write this on your paper. In DNA, the bases are going to be complementary, meaning they're going to pair up in a predictable way. And it's like a zipper. If you've ever zipped up your sweater, you know that the teeth are going to stack up in a predictable way, which then causes the sweater to close or open. And any sort of defect or mess up in the zipper strand means it's not going to zip up like you should, and that's the same thing with DNA. So in DNA, on either side of the zipper, the bases are going to pair up in the following way. It's going to be adenine with thymine, and the way I remember this is apple tree. Apple tree, adenine and thymine always pair up. The second pair is cytosine and guanine, chewing gum. Apple tree, chewing gum. Those are how they pair up. You're never going to have adenine and guanine, cytosine and thymine. It's always going to be apple tree, chewing gum. You don't have a chewing tree and you don't have apple gum. Okay, apple tree, chewing gum. Now in RNA, what makes it stand out from DNA is that in RNA, you do not have the base thymine. So it's not going to be apple tree anymore. It's going to be adenine pairing with uracil. So the uracil is going to replace the thymine. And that's what's going to distinguish RNA from DNA. Okay, so here's an example of what a DNA nucleotide would look like, an RNA nucleotide would look like, and then the 4 individual bases that occur in the DNA. Okay, so let's talk a little bit about DNA specifically. So DNA is a double molecule, or called a double helix. It resembles a twisted ladder, so you kind of take a ladder with its rungs, its little stair steps, and then you twist it, and that's what DNA looks like. It's a coil. There are sugar and phosphate groups making up the backbone. The 2 strands are going to be linked by nitrogenous bases to make the stairs, and that's where you're going to have your complementary bases.
Can we go now?